Good afternoon, everybody, and welcome back. We're going to resume the program with the afternoon session. There'll be 4 talks this afternoon, and there will not be a break, so we'll go straight through to the end. Our first speaker is Jackie Lees. As you heard from Jackie's introductory remarks and her accent, she grew up in Brooklyn. It's not true. She grew up in England. And she got her PhD at the Imperial Cancer Research Fund, which is now called the London Cancer Institute in the United Kingdom. And from there, she came to Boston and did her postdoctoral studies with Ed Harlow, actually started at Cold Spring Harbor and moved to Boston with Ed, where she did groundbreaking work on the E2F family of transcription factors and their regulation by RB and other pocket proteins. We then recruited Jackie to the Koch Institute, then the Center for Cancer Research, where she's continued to do very important work in the area of tumorigenesis, cell cycle control, and models of cancer, both in the mouse and in zebrafish. Jackie is now professor of biology, a Daniel K. Ludwig scholar, and importantly is the associate director of the Koch Institute. And she's going to tell us today about work from her lab involving BMI1, stem cells, and cancer.
Thank you, Tyler. So as Tyler mentioned, I'm going to talk to you today about an unpublished story looking at the role of BMI1 in intestinal development and tumorigenicity. And I want to start right out by giving credit to Mindy Maynard, who's the highly talented postdoc in the lab who's really responsible for almost entirely for the work that I'm going to show you today. And this subtitle is actually Mindy's favorite title for this talk. So as Tyler mentioned, I'm going to tell you today about BMI1. And BMI1 is actually a component of the PRC1 epigenetic regulator complex. And this cartoon just shows that PRC1 actually binds to chromatin modifications that are laid down by PRC2. Once it's associated with those, it then carries out ubiquitination of H2A, lysine 119, and mediates transcriptional silencing. And BMI1 regulates a large number of genes, but the 3 that are best understood are these proteins here. So P19ARF, the tumor suppressor that's a key upstream regulator of P53, P16, again, a tumor suppressor that's a component of the RB protein, and P21, a downstream target of P53. And collectively, these 3 proteins are capable of mediating arrest, senescence, and apoptosis. Now, consistent with BMI1's role in inhibiting these activities, BMI1 is a well-documented oncogene. It was originally identified by Martin van Looijen as an oncogene that cooperates with MYC in lymphomagenesis. But subsequent studies then showed that BMI1 is highly expressed in a wide variety of human tumors, firstly hematopoietic lineages, but also many, many solid tumors. In fact, pretty much every tumor that's been looked at has elevated BMI1 expression. And what's most important is that this high BMI1 expression correlates very well with poor prognosis. And this just shows you an example of BMI1 expression in colorectal cancer, which is the topic of my talk today. So a number of years ago, Martin van Looijen's lab made a BMI1 germline mutant mouse, and that's really provided key insight into BMI1's role in a variety of tissues. So it turns out that these mice have severe defects in the hematopoietic and neuronal compartments. And in both of these cases, these defects result essentially from loss of self-renewal and proliferative capacity of the stem cells, and that's related to the upregulation of the 3 regulators I just told you about, P19ARF, P16, and P21. Now, not surprisingly, since these compartments are impaired, it was shown that these mice are also highly resistant to transformation of the hematopoietic and neuronal compartments. Subsequent studies looked at breast, and the breast in these mice is essentially normal, but if you transplant the stem cells, you see very clearly that there is ensuing stem cell exhaustion in these populations. And if you look at transformation of those cells, you find that transformation is also greatly impaired. And then a number of years ago now, in a collaborative study with Carla Kim's lab, my lab looked at the lung and showed that the lung is essentially normal in BMI1 mutant mice. But again, if you damage the normal lung, you see evidence of stem cell exhaustion. And in this setting, BMI1 is really critical for transformation of the lung by the KRAS oncogene. So the take-home message here really links BMI1 to stem cell capacity, both in a variety of normal tissues and also tumorigenicity. So we wanted to look at BMI1's role in intestine, both because of the high level of BMI1 in intestinal tissues, and also because it had been well demonstrated by a number of labs that BMI1 is highly expressed in intestinal stem cells. But surprisingly, papers have reported that the intestine is completely normal in BMI1 deficient mice. So what we've done is to take a mouse model that's driven by loss of the APC tumor suppressor. And just to remind you, APC is frequently inactivated in a variety of, in a large proportion of human colorectal cancer, and that's an initiating mutation. And what APC does in normal cells is to repress Wnt beta-catenin signaling, which is required for intestinal stem cell maintenance. Now, this mouse model carries one floxibly allele of APC, and that's inactivated by vilin-Cre, which is highly specific for the intestine and is also highly penetrant. These mice then undergo LOH of the remaining allele, upregulate signaling pathways, and they get a large spectrum of intestinal adenomas very rapidly. So what we've done is to cross the BMI1 mutant mice, germline mutant mice, into this model and compare the tumor phenotypes of mice that are wild-type heterozygous or homozygous mutant for BMI1. And what you can see very clearly is that there's actually very profound suppression of the tumor phenotype in these animals. So this shows you histological sections from the small intestine of mice that are 120 days of age. And you can see that there's major tumorigenic lesions in the BMI1 mutant mice. And the intestine, at least at the gross level of the BMI1 null mice, looks essentially normal. And that follows with quantification. If we look for visible adenomas, we basically have never seen a visible adenoma in the BMI1 null animals, whereas wild-type controls typically have in the order of 80 to 90 tumors. And what you can see very clearly that the heterozygous mutant state gives an intermediate phenotype. So BMI1 is acting in this tissue in a dose-dependent manner. We do see microscopic lesions in these mice, but they are significantly smaller in the BMI1 mutants than the wild-type controls. And I just want to point out that this is a log scale, so there's an order of magnitude difference here. And finally, Mindy's shown that BMI1 loss also suppresses large intestinal tumors in this system. So Mindy wanted to look at how this phenotype changes over time. And what she did is to actually step back to an earlier time point, 90 days, and carry out the same type of analysis and compare it to the 120-day phenotype. And the data are shown here. So in wild-type mice, what you see is that there's absolutely no difference in the number of lesions that you get between the two time points. But there's a significant increase in lesion size, consistent with the idea that these tumors are progressing rapidly in this time frame. Now, we see something very different in the BMI1 mutant animals. Firstly, the tumors are equally small at both the 90 and 120-day time point, so there seems to be no progression between these two phases. And secondly, we were quite surprised to find that there's actually more tumors present at 90 days than there are at 120 days. And these data actually argue that there's two things going on here. Firstly, that BMI1 deficiency suppresses developing tumors at a very early stage. And secondly, that the BMI1 deficiency is actually promoting clearance of tumors that existed at earlier time points. So Mindy's really worked primarily on the cause of this early stage defect. And first off, she wanted to verify that, in fact, BMI1 is not interfering with tumor initiation. And she was able to show very clearly that BMI1 loss does not affect the rate of recombination or loss of heterozygosity of APC or activation of signaling of beta-catenin or c-MYC. So having established that initiation occurs at normal, she then looked at other events, and particularly she focused on the three regulators that I've already told you about as being strongly implicated in BMI1's tumor suppressive activity. Now, the BMI1 null tumors are too small to carry out Western blotting analysis, but Mindy made extracts from tumors that were either BMI1 wild type or heterozygous. And what you can see very clearly is that even mutation of a single allele of BMI1 allows upregulation of P19ARF in these tumors, And in contrast, we see no effect on P16. So that argues very strongly that inactivation of BMI1 is upregulating P19ARF, and Mindy then wanted to assess these known downstream consequences. And she started off by looking for a rest, because as I've told you already, the loss of proliferative capacity is really the hallmark of BMI1 mutation, both in normal stem cells and in tumor tissues. And given that, so what we did is to do BRDU incorporation, and here's the wild-type tumor and the BMI1 null tumor. And what Mindy was very surprised to find is that, in fact, there's a significant upregulation of proliferation in the absence of BMI1. And that's actually true even if you stratify the tumors based on size. So what our data are arguing is that BMI1 loss is actually somehow increasing proliferation, and I'll come back and tell you in a little while why we think that is, and that therefore strongly rules out arrest as being the causative effect in this tumor suppressant. Now, Mindy's also looked at cellular senescence, and she's shown that there's a very similar level of senescence in mice that are either wild type or mutant for BMI1. So that did not seem to be the cause of this tumor suppression, and that really left apoptosis. And consistent with this observation, what Mindy was able to show is that in the BMI1 null tumors, she sees a significant activation of apoptosis in the null state. And what's very important in this observation is that this apoptosis was absolutely specific for the very smallest lesions, one that have a cross-sectional area of less than 10 to the 4 micrometers squared. Once the tumors get larger than that, she sees no induction of apoptosis in the BMI1 null state. So this actually supports two conclusions. Firstly, we believe that inactivation of BMI1 is leading to apoptosis through the upregulation of P19ARF. And secondly, the fact that this is specific to the smallest lesion provides a strong suggestion that, in fact, there must be some compensatory mechanism as the tumors progress that wipes out this apoptosis and allows the tumors to progress to this larger phase. And consistent with this hypothesis, Mindy looked at the tumors more carefully, and what she was able to see is that the BMI1 null tumors have a very high level of nuclear P53, which is essentially analogous to mutant P53. And what she found is that approximately 40% of the cells in the tumor on average were high for P53. So this is really arguing that, in fact, what's happening here is that we're getting induction of P19ARF and apoptosis, that this creates very strong selective pressure early on for P53 mutation. That feeds back, suppresses the apoptosis, allowing the tumors to progress. And we believe that the loss of P53 in this system is also accounting for the increase of proliferation that we see in our BMI1 null tumors. Now, if this model is correct, what we would predict is that mutation of ARF in this model would significantly reduce all of these discovered phenotypes. So to test this hypothesis, Mindy crossed an ARF mutant allele into her tumor model and then assayed for these defects. And it's exactly as you would have predicted. What Mindy found is that in tumors that were doubly deficient for both BMI1 and ARF, she now completely eliminates the apoptosis in the small regions. She also completely eliminates the accumulation of mutant P53 in these tumors. And these data argue very strongly for validation of the hypothesis that I've shown here. But it also allows us to address the degree to which these events, the upregulation of ARF, the induction of apoptosis, might be contributing to the tumor suppression that we see in the BMI1 null state. So Mindy's taken the double mutant animals and now compared their tumor phenotype directly to those of mice that were either wild type or deficient only for BMI1. And the data are shown here. So this again is section from 90-day-old animals. Here's the BMI1 wild type, lots of tumors, BMI1 deficient animals looking with no visible tumors. And here we see the double mutant, and you can see clearly now that there is some increase in the tumor phenotype in these mice. But what's important is that this increase is extremely subtle. We see only a very low number of visible tumors, and I should point out again, this is a log scale, so we get really 1 or 2 tumors relative to the 90 or so in the control animals. We also see an increase in both the number and the size of microscopic tumors, but again, this does not approach the level that you see in the BMI1 wild-type animals. Okay, so to conclude this part of my talk, what I've shown you so far is that BMI1 loss profoundly suppresses intestinal tumorigenicity. I've shown you that BMI1 loss induces apoptosis in early lesions, and this apoptosis is ARF-dependent. And I didn't have time to tell you the data, but I can tell you that this defect really reflects a cell-autonomous defect in the intestinal epithelium. And Mindy achieved that by generating, with support of others, a conditional BMI1 mutant allele that was deleted along with the APC inactivation with Villin-Cree. And what you can see here is that the mice with the conditional alleles of BMI1, so BMI1 deleted only in the intestine, have essentially exactly the same degree of early tumor suppression as you see in the germline BMI1 mutant mice. So this is really a defect of the intestinal epithelium. Now, despite this early tumor suppression, our data argue very strongly that there's actually at least one or more additional mechanisms that are able to suppress intestinal tumors in the absence of BMI1. And this is capable of restraining the tumors that have had mutations of ARF, either introduced by us or p53 mutations that arise spontaneously in the mutant animal. And I also want to remind you of the point that I made earlier, that BMI1 loss promotes tumor clearance in these animals from 90 to 128 days. And this is raising the possibility in our mind that this could reflect the defect in the hematopoietic compartment that's present in the BMI1 mutant animals, because it's well established that there's an important effect of inflammatory response in promoting intestinal tumorigenicity in this system. And we believe we may have actually modulated this in part by disruption of the hematopoietic compartment, and Mindy's currently testing that. Okay, so I've told you so far about BMI1 in the tumor phenotype, but now I want to switch gears and tell you a little bit about BMI1's role in the normal intestine. And I have to remind you that this story was a big surprise to us because many of the previous papers had argued that the intestine was completely normal in BMI1 mutant mice. But when Mindy was looking at the BMI1 mutant animals in the context of this tumor model, she also realized that adjacent epithelium, one that looked completely non-transformed, really didn't look exactly right in these animals. And therefore, she went on to test BMI1's role in normal intestine, now using models that lack either the APC, lack inactivation of the APC tumor suppressor. So before I tell you these data, I just need to give you a summary or introduction to intestinal homeostasis. So this is a picture of the villus in the intestine. Here's the lumen. And what's important to note is that the villus is predominantly comprised of terminally differentiated cells. And the vast majority of these cells is an absorptive cell lineage called enterocytes. But there are also scattered amongst its secretory cell populations, which are goblet cells and enteroendocrine cells. Now, these cells arise from the differentiation of stem and progenitor cells that are actually contained within the intestinal crypt. And in the intestine, there's actually 2 distinct stem cell populations that have been identified. The first is the +4 stem cell, named because of the position it sits above the base of the crypt. This cell is essentially quiescent in the normal intestine, but it's capable of self-renewal, and also in certain circumstances is capable of giving rise to these LGR5 stem cells. The LGR5 stem cells sit at the base of the crypt. There's actually multiple of them. They're supported by Paneth cells, which is a secretory cell lineage that's important for this niche. And these cells are really much more proliferative, and in the normal situation, they're responsible for producing the progenitor cells that comprise the rest of the crypt. And importantly, these progenitor cells can undergo differentiation either to produce the absorptive lineages up here or to produce secretory cells, including the endocrine cells, the goblet cells, and the Paneth cells, which migrate back down to the bottom of the crypt. Now, early work from the Kopecky lab argued that the +4 stem cells, this quiescent stem cell, was the only cell in the intestine that had BMI1 expression. But work from others has subsequently shown that BMI1 is expressed in the LGR5 stem cells, albeit at somewhat lower levels. And collaborative studies we've conducted in collaboration with Shalev Itzkovitz in Alex van Waarden's lab has shown that, in fact, these progenitor cells also express BMI1, and this was done using single molecule transcript analysis. And I'll return to the point of this in a moment. Okay. So Mindy's looked hard at the intestine of the BMI1 deficient mice, and she realized that there were actually two obvious defects here. The first one is that BMI1 loss is actually causing significant apoptosis of the absorptive enterocytes. And this apoptosis is actually specific to the cells that are towards the base of the crypt, but they're terminally differentiated, and that's demonstrated here looking for staining for cleaved caspase 3. So this apoptosis is systemic. It seems to be occurring in real time, but does not affect grossly the structure of the intestine. Now, since we'd shown that upregulation is responsible for the apoptosis in the intestinal tumors, Mindy wanted to ask how ARF affects this apoptosis, and we were quite surprised to find that, in fact, this apoptosis in the normal intestine is entirely ARF independent. There's essentially exactly the same level of apoptosis in the single versus the double mutant animals. The second defect that Mindy sees is actually a defect in the specification of cell lineages. And in particular, she sees underrepresentation of two of the secretory cell lineages, Paneth cells and goblet cells. And that's shown here. So remember, I told you that the Paneth cells sit at the base of the crypt, and you can see that there's far less staining in the BMI1 null animals versus the BMI1 wild-type controls. This is in the small intestine. Similarly, if we look at goblet cells, which are present both in the crypt and in the villi, you can see again that there's more goblet cells present in wild-type animals in both the small intestine and the large intestine than there are in the BMI1 mutant mice. And this is actually just quantification of that defect. Mindy actually counted the representation of cells per crypt unit, taking a similar positioned section in the two mouse models. And you can see here that all other lineages are present at normal levels, but there's significant underrepresentation of the goblet and Paneth cells. So what's the cause of this defect? So I want to remind you again that I told you that BMI1 is expressed in both stem cell populations and also the progenitor cells. So it seemed that each one of these could be a potential source of the defect. And we initiated studies by looking at the LRG5+ stem cells for two reasons. One is because I've told you in every other setting, BMI1's function seems to be particularly important in the context of stem cell maintenance. And secondly, I've told you that we have a reduction in the number of Paneth cells, and the Paneth cells are known to be an essential niche to support this stem cell population. So to look at these cells, Mindy crossed the BMI1 mutant mouse model into a reporter mouse in which the LRG5 promoter is used to express GFP. She then FACS-sorted cells out that were either BMI1 wild type or BMI1 mutant, placed those cells in culture, and looked at their ability to form organoids in this in vitro assay. And what we were surprised to find is that the BMI1 mutant cells do this very efficiently. And in fact, in 3 independent experiments, these cells consistently do it better than the wild-type population. So this argues very strongly that BMI1 loss is not impairing the ability of the LRG5+ stem cells, at least at this time point of development, and therefore argues against this being the cause of our defect. So Mindy then turned her attention to the progenitor cells, and I've told you that progenitor cells are capable of differentiating either into the absorptive or the secretory lineages. And this is carried out under the control of Notch signaling. So what happens is that Notch is responsible for activating HES1, and HES1 is a transcriptional repressor that blocks another transcription factor, atonal 1 or MATH1, that's essential for commitment to the secretory lineage. Once cells are at this point, they can then differentiate either to form enteroendocrine cells or select Paneth and goblet cell lineages, and that's under control of another transcriptional regulator, GFI1, that's regulated independently of Notch signaling. So Mindy started out to look at the expression of, and I should point out that work from Christian Hellen's lab using knockdown of BMI1 in tissue culture cells had shown that HES1 can actually get upregulated in the absence of BMI1. So Mindy looked at the level of these factors, initially by Western blotting, and the way that she did this experiment is to actually dissect out a crypt-enriched region of the epithelium, make cell extracts, and then Western blot in the wild type versus the mutant epithelium. And you can see in this experiment, in the BMI1 now animals, we have upregulation of HES1, downregulation of atonal 1, and that's consistent with the idea that HES1 is a negative regulator of this gene. And Mindy saw also that there's downregulation of GFI1, the factor that influences choice between the Paneth and goblet and enteroendocrine cells. And the downregulation of this factor is very consistent with the fact that we're selectively losing the Paneth and goblet cell lineages. So Mindy wanted to look more carefully at where these transcriptional changes were occurring. Are they really related to the progenitor cells? And to do that, again, with the help of Shalev Itzkovitz and Alex Van Waarden, they conducted small molecule transcript analysis in the crypt for HES1, which is shown in red, and atonal 1, which is shown in green. And I'm not sure if you can see it out there because I'm having a hard time seeing it. But what she found is that HES1 is actually upregulated in the BMI1 mutant, so there's an increase in red and downregulation of the atonal green marker. And importantly, these changes affect both the number of cells that are expressing the transcript and actually the number of transcripts present per cell. Okay, so in the second part of my talk, what I've told you is that contrary to the published view, BMI1 is actually required for normal intestinal homeostasis. I've told you that BMI1 losses induces apoptosis of the absorptive enterocytes, and that this defect seems to be entirely independent of ARF. And I've told you that BMI1 loss also is required for the commitment of the Paneth, commitment of the crypt progenitors to give rise to the Paneth and goblet cell lineages. Again, I didn't have time to show you the data, but Mindy's looked in the conditional mutant mice, and this is really an intrinsic defect in the intestinal epithelium. And we believe that this occurs through modulation of Notch pathway and GFI1 proteins. So BMI1, we think, is inhibiting HES1, inhibiting GFI1, and that does not rule out the possibility that we're currently analyzing that BMI1 might talk through other mechanisms to Notch signaling. So I just have one more slide that I want to show you, and I really want to bring this back to the big picture and remind you that we started this work because BMI1 is highly expressed in a wide variety of human tumors, and that, again, this correlates with poor prognosis. So I've already told you about the data in these 4 different tissues, hematopoietic, neuronal, breast, and lung, and that in each of these settings, BMI1 seems to be critical for tumorigenicity and either have either moderate or strong effects on development. And all of these defects seem to be related to the requirement of BMI1 to maintain self-renewal and proliferative capacity of the stem cells through its ability to repress ARF P16 and P21. What we've done now is to add intestine to the list, and I think what's important to note is that this has the same general trend. There are subtle defects in development, and there's a great impairment of tumorigenicity in these models, this tissue, but this is occurring through distinct mechanisms than these other tissues. We see no evidence of stem cell defects, at least in the normal situation, and instead we have apoptosis of differentiated cells and specification defects within progenitor populations. In the case of the tumorigenic model, we have impairment, we have upregulation of ARF and apoptosis in early lesions, but there's also additional mechanisms that appear to be entirely ARF independent in this model that we're actively pursuing. And I think as a take-home message, this really raises interesting ideas about whether or not BMI1 might be a good target for tumor treatment. What you can see is now there's a growing list of tissues in which BMI1 is required at a very early stage for tumorigenicity. through a variety of mechanisms. But there's also an impact of BMI1 loss on development that I think we need to understand better. I think the most encouraging thing is that we've in fact shown that in the heterozygous mutant state, BMI1 heterozygosity impinges upon tumorigenicity, but has no effect on normal development. And that argues that we should be able to titrate a level of BMI1 downregulation that maybe could really be directed towards the tumorigenic setting and not be too harmful on the normal situation. And I just want to end by acknowledging again Mindy, who, as I said, did almost all of this work. The exception was that she generated and characterized the conditional mutant mice with help from Roberta Ferretti in my lab and also Peter White, who at the time was on sabbatical in my lab. The single molecule transcript analysis I showed you was work of Shalev in Alex's lab. And I really have to acknowledge the KI Swanson Biotechnology Center and all these really outstanding cores, transgenic histology, pathology, and flow cytometry that were critical for this work. And I'll thank you very much for your attention.